competitive botany. In this video, you are going to learn about the nucleus. Nucleus is also called as karyon, was first discovered by the scientist Robert Brown in the year 1831 in the epidermal cells of orchid roots. So the nucleus is called as dynamic center of the cell. It is also described as master control of the cell. It is also commonly called as cell brain because it controls all metabolic activities as well as hereditary activities of the cell as it contains the DNA. So here you can see the true nucleus. So see the picture carefully. It is a true nucleus. That means a true nucleus present in all eukaryotic cells, nothing but it has a distinct nuclear envelope around the nucleus. So it is only present in eukaryotes. Okay. So you can see the nuclear envelope here. Okay. So while in the case of prokaryotes, there is no envelope around the nuclear material. That means there is a naked nuclear material is present. So that's why they are called as prokaryotes because they consist a primitive nucleus. So in this picture you can see the naked chromatin material. Cells with the single nucleus is called as monokaryotic cells, whereas the cells with two nuclei are called as dikaryotic cells. So example, members of Ascomycetes and Basidiomycetes of fungi contain the dikaryotic cells. That means they contain two nuclei in their cells. So let's discuss the structure of the nucleus. So generally nucleus consists the four major parts. It consists the four major parts. They are nuclear envelope, nuclear sac or nucleoplasm, chromatin material and nucleolus. Let's see one by one. So here the first one is nuclear envelope. Here you can see the blue color envelope present around the nucleus. Okay, so in the diagram it is represented as blue color, okay, for your convenience. Okay, so which is a nuclear envelope, so that is present around the true nucleus. Okay, the nuclear material that is separated from the cytoplasm by an envelope is called nuclear envelope. It is also called as karyotheca. So it consists of two unit membranes. The space present between the two membranes is called as perinuclear space. So the outer membrane is always associated with the endoplasmic reticulum. Okay, so next, at regular intervals in the nuclear envelope, it consists of pore-like structures. So the diameter of each pore is 400 angstroms. The diameter of each pore is 400 angstroms. So nucleocytoplasmic transport of materials is carried out through these pores. So that means the materials that are carried out of the nucleus occurs through the nuclear pores. So the nuclear envelope actually disappears at the end of prophase of the cell division. You should remember. So next, coming to the second part, that is nuclear sac or nucleoplasm. So it is also called as karyoplasm or karyolymph or nuclear matrix. You can see the picture carefully. So nucleoplasm that is present inside the nucleus. So intranuclear protoplasm is called as nucleoplasm. Nuclear membrane encloses a fluid-like non-staining region which is called as nuclear sap or nuclear matrix. It includes nuclear sap or nucleoplasm includes chromatin reticulum, nucleoli, enzymes, nucleotides and also proteins. Let's see the third part, chromatin material. So it is present in the nucleoplasm. See the picture. So these black thread-like structures are called as chromatin material. Okay. So the nucleoplasm contain many thread-like coiled stained bodies. They are called as chromatin fibers. So if they are deeply stained portions are called heterochromatin. If they consist, the heterochromatin generally consists inactive genes or non-functional genes. So the remaining portion of the chromatin that takes little stain are called as euchromatin. Should remember, euchromatin consists structural genes or functional genes. Chemically, chromatin consists of DNA and histone proteins. DNA and histone proteins. There are 5 types of histone proteins known as H1, H2A, H2B, H3 and H4. So the histones are the basic proteins. That means they consist a basic nature because they have high content of basic amino acids such as arginine and lysine. Should remember. So being basic, they are bind tightly to the DNA because DNA have the acidic nature. So during the cell division, chromatin fibers become thick and ribbon-like. They are called as chromosomes. So now coming to the fourth part, that is the nucleolus. See the picture carefully. Nucleolus is also called as plasmosome. It is a deeply stained spherical body present in all true nucleus. So it is composed of RNA, proteins and little amount of DNA also present inside the nucleolus. So here you can see the blue round circle like structure present inside the nucleus. So the number of nucleolus per cell is different in different organisms. So the nucleolus is synthesized basically from the nucleolar organizer region NOR, such as secondary constriction region of SAT chromosome. So the nucleolus plays an important role in the production of ribosomes. Hence it is called as ribosomal factory or seat of ribosomes. It plays an important role in cell division. Usually it disappears at the end of prophase and reappears at the end of telophase. Okay, so this is regarding the structure of nucleus. So now we are going to deal the important competitive bits that are from nucleus. Let's see one by one. The study of nucleus is called as, answer is karyology. Nucleus discovered by the scientist Robert Brown. The nucleus shows all functional activities of the cell and it controls all cellular activities as I told you before. So nucleus is concerned with ultimate control of cell activities. True nucleus is absent in, answer is bacteria. Nucleoids are found in bacteria and blue-green algae. So as I told you before, the prokaryotes contain the naked chromatin material of that is also called as naked chromatin material, that is also called as nucleoid or xenophore. So chromatoplasm is found in the cells of, answer is blue-green algae. So chromatoplasm is nothing but the peripheral protoplasm of blue-green alga, which contains generally chlorophyll and accessory pigments together with stored substances. So that is present in blue-green algae. The boundary between nucleus and cytoplasm is, answer is nuclear envelope. So the term cytomembrane or endomembrane given by the scientist, answer is D. Robertis. So endomembrane or cytomembrane is nothing but nuclear membrane, endoplasmic reticulum, Golgi complex are together called as endomembrane or cytomembrane. Generally, they are joined together to form endomembrane system of the cell. You can see the picture carefully. So the nuclear envelope is connected to the endoplasmic reticulum. The endoplasmic reticulum is connected to the Golgi complex. Then the Golgi complex is also connected to the plasma membrane. So which is that forms an endomembrane system inside the cell. Okay. So next. Nuclear membrane facilitates nucleocytoplasmic exchange of materials. Okay. Next question. Cellular structures which always disappear during cell division are nucleolus and nuclear membrane. Both are disappear during the cell division. Okay. Next question. The main substance involved in transmission of characters from cell to cell is, answer is chromatin. DNA is concentrated in chromatin. The term chromatin was coined by, answer is Fleming. The term heterochromatin was coined by, answer is Emil Higgs. Cell component which transmits genetic information from one generation to the other, answer is chromosome. Answer is chromosome. The term chromosome given by the scientist, answer is Waldeyer. Nucleolus was first observed by, answer is Fontana. The term nucleolus given by the scientist Baumann. So generally nucleolus is made up of rRNA and proteins. It consists a little amount of DNA also. Chief role of nucleolus is ribosome synthesis. Site of formation of immature ribosomal subunits in eukaryotic cell is, answer is nucleolus. So next, which structures are called units of chromosomes? Answer is nucleosomes. Nucleosomes are called as units of chromosomes. So here you have to know what are nucleosomes. See the picture carefully. Chromatin material consists of repeating units called nucleosomes. Under the electron microscope, the chromatin fibers appears to have a string of beads-like structures. So these beads were called as nucleosomes. Here you can see the bead-like structures, nothing but nucleosomes. Next question. An octamer of 4 histones complexed with DNA is called as nucleosome. As I told you before, there are 5 histone proteins: H1, H2A, H2B, H3 and H4. Generally, H2A, H2B, H3 and H4, they form an octamer. Around this octamer, the DNA is wrapped. Like you can see the picture. The DNA is wrapped around the octamer of histone proteins. So that each round octamer is called as nucleosomes. Okay. So let's see the next question. These nucleosomes are linked together by, answer is DNA. A nucleosome consists of 200 base pairs of DNA. The role of nucleus in regulating the morphology of plant was discovered in, answer is Acetabularia. So these experiments have made by the scientist Hammerling. Hematoxylin is used to stain the nucleus. The molecules that move regularly nucleus and cytoplasm. The molecules that move regularly from nucleus to the cytoplasm are mRNA and nucleoproteins. Which cell division stage is ideal stage to study the structure of nucleus? Answer is interphase. Answer is interphase.